Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. About a year ago, I had the chance to go on to the Degrees of Health podcast with a guy named Ben out of the UK who's doing great work and having great guests on and having great conversations that I personally enjoy. But Ben was nice enough to hook me up with the contact information to a Professor Thomas Seager, who is the CEO and co-founder of Morozko Forge ice baths. And even though I completely screwed up the intro, you'll see what I mean in a few minutes here. But I had a great conversation with him. It was a very interesting conversation. It covered a ton of different topics, some of which I was expecting, some of which I was not expecting. The conversation lasted roughly 2 hours. I even had a chance to talk to him offline for about 30 or 40 minutes after the interview was over. And given his background in engineering as a professor of engineering, it was really interesting to get his perspective from his view on health and disease. I ended up breaking the podcast into two parts. I'll drop this podcast here and then I'll have another one next week because it was so long. But in this part, we talk about mitochondria, we talk about biophotons, how cold affects the body at the mitochondrial level, and how cold may even stimulate vitamin D synthesis inside the body. Pretty cool. I hope you enjoy. I have the pleasure of having Dr. or Professor Thomas Seager on the show today, who is an associate professor of sustainable engineering, or excuse me, at the Arizona School of Sustainable Engineering at Arizona State University in Tempe, Arizona, but is also the CEO of Morozko Forge, which he co-founded in 2018. And I just want to say thank you so much for being on the show, Professor.
Thanks for inviting me, Dr. Peavler. It's an interesting distinction. People throw this word doctorate around because it means, I guess, you have a doctorate, but I'm a professor. And my expertise started in engineering. And I don't want anybody to think that, I could prescribe them a GLP-1 or anything. I build machines.
Yes, that's that. I think that's probably what makes your story very interesting because you are this professor of engineering and you have transformed into this authority on cold water immersion and became this again CEO co-founder of Morozko Forge. And it's funny because I had learned about cold water immersion because I've been following Dr. Cruz for so many years. Like I right out of residency, I was in 2017, I was able to watch that podcast that he did in Vermont. And it's kind of one of his more famous podcasts. And he was talking about how he lost all this weight using cold water immersion. At the time, in that time, because he was talking about like 2012, 2013, there was no Morozko Forge. There was no cold plunges, right? So he was using just like ice in a bathtub and he had a whole protocol that was built. This was way before all the devices were made. So like the actual process came on my radar then. And then I think the first I was introduced to you was back whenever it was in those days when Ben Greenfield did a podcast with you and he was showing off his Morozko Forge in that podcast. I'm like, okay, let me check this out. They make plunges now. So anyway, how did it go from professor of engineering when I've heard all you do this cool stuff in Cartagena and working with the Army Corps of Engineers to building your own plunge? Like what led to that?
Necessity, they say is the mother of invention. And that was pretty much the case for me too. My head was filled with all kinds of wrong ideas about nutrition, about metabolism, and about health. And one of the things I feel most terrible about is the way that my son has borne the consequences of those wrong ideas. When he was 6 years old, he was diagnosed with type 1 diabetes. I didn't know what was wrong with him. It was maybe a year after we started insulin injections and testing his blood sugar several times a day. I think I was sitting in a dentist's office and I picked up a copy of Reader's Digest because, back then they still had those things. And it was talking about a study that came out of Finland, took them 20 or 25 years to do on type 1 diabetes. This is a longitudinal study. So they were tracking pregnant women and their children going all the way back decades ago. I think they started this in the 70s. They discovered The diabetes originates in vitamin D insufficiency in the first year of life. You know, and but for the audience's benefit, a child is born into this world, a baby, it does not have a mature immune system. It is depending upon its mother's breast milk for the antibodies that it needs to stay healthy. And it is amazing the relationship between the mother and the child because the composition of the breast milk will adjust to meet the child's needs as it is developing its own immune system. Vitamin D is essential to the development of that immune system. And what the Finnish longitudinal study discovered is that if the mother and the child are deficient in vitamin D, the risk of this autoimmune disorder, which is type 1 diabetes, the risk of a type 1 diabetes diagnosis goes way up. I didn't know any of this when my son was diagnosed, but as I replayed it in my mind, I realized he was born in late October. We were living in northern New York. There's no way he
No
got
way.
enough vitamin D. There were friends of mine, colleagues that had two sons. I was way up in Potsdam, New York at the time, and both of their sons were also diagnosed with type 1 diabetes. Why did the Finns even do this study? Because when you live at those extreme northern latitudes, of course, the days are very short in the winter, and people don't get enough sunshine. Even if they are getting sunshine in the winter, of course, enough UVB to stimulate vitamin D production. I only found out later, and Jack Cruz and Alexis Cowan helped a lot with this. I only found out later that cold can compensate for a lack of sunshine. When you get in the cold, it activates your brown fat. So these are the mitochondria in your brown fat, and they are producing heat from mostly fats, but a little bit of glucose gets in there. It's oxidized, and instead of producing ATP, they produce warmth to keep your core body temperature up despite the fact that you're in the cold. That mitochondrial activity produces what are called now biophotons. The electron transfers in the mitochondria cannot help but produce light. That's the way these redox reactions work. So they're producing light inside your fat cells. Some of that light is in the UVB range. When that UVB wavelength, which they call ultra weak because it's so difficult to detect, but when it is produced in the mitochondria and it intersects a cholesterol molecule, it will convert that cholesterol to pre-vitamin D that is then later converted into any of the 25 different forms or something like this of vitamin D in your body. Now, vitamin D is stored in the fat cell. So when you produce the light in the body and you produce the vitamin D in the fat cell, it need not go anywhere. It can stay right in the fat cell because it is so lipophilic until your body needs it somewhere else and then it can enter your bloodstream. So there's really no reason to think that if you're producing vitamin D via cold exposure instead of in your skin via sunlight, that your serum levels of calciferol or whatever form of vitamin D you're testing for should increase, at least not right away. But a
Yeah,
new
that's
study
interesting.
came out.
I remember you talking to Max Gulhane about this on a
Yep.
podcast, and who I also really like following him because he's also a Dr. Cruz fan, and he's had Cruz on to his podcast a couple of times. I remember you telling me about this. What my understanding though is that, and at least what I've heard Dr. Cruz say about a billion times is that we don't have the photomultiplier intracellular photomultipliers to see exactly what those biophotons are. We know it's happening because of, like, I don't remember what the guy's name was. It's not Roland Van Wyck, but one of the guys he always talks about. And they were saying that, like, I know I've heard Dr. Cruz say this. He hypothesizes they are in the UV range, but we're not sure exactly what wavelength or not. I think it's an interesting thing, and Max brought up the point, that that IR, will make mitochondria make their own melatonin, which we know about. Cancer cells have that mechanism turned off, interestingly enough. But that being said, so I think it could make sense. It's just that it's like you said, it's hard to prove because it's hard to know. But I think everybody. I mean, I've heard Alexis say this. I've heard, of course, Dr. Cruz say this about a zillion times. I think it is very likely true. But you also made a point during
There's
that
been a lot
podcast.
more since I talked to Max about it. There's
Interesting.
been new studies. Now,
Interesting.
outside the body, these ultra weak biophotons are very difficult to measure. Of course they are, because they're made on this microscopic scale. And although IR, infrared, will penetrate the tissues of the skin, UV will not. They
Right.
don't travel very far in the body. And so, what probe, what sensor, what detector are you going to use to find the UV wavelengths produced by the mitochondria outside the body? This is a technical challenge.
Oh, yeah.
But the photomultipliers that will amplify that weak signal are getting better and better. And this is just in the last 2 months. Suzanne Humphreys drew my attention to this with a tweet that she put out. Biophotons have now been observed in every living system examined. So, this is a new paper that she helped summarize that said, look, biophotons are life. They compare the living tissues and the dead tissues. It doesn't matter, plant or animal, they all have mitochondria. And when you compare a leaf or a mouse that is living to a leaf or a mouse that is dead, you can see the difference in the biophoton production. This is not a matter of speculation. This is a matter of physics. You cannot move an electron from one shell to another. You cannot have a redox reaction without the release of light from that electron movement. So there are a few steps in the chain that are very reliable. We know the light is released. We know some of that is in the UVB range. We know that the cholesterol is sitting right there next to the locus of biophoton generation. We know that some of that light is going to intersect that cholesterol. And we know, again, from physics, that that cholesterol will be metabolized, not metabolized, it will be converted to pre-vitamin D. That the next thing is, what happens after that?
Right.
The pre-vitamin D doesn't just go away. The conversion of pre-vitamin D into the first form of vitamin D is not mediated by any enzyme or biological process. It happens, I'm going to say mechanically, but in response to warmth, the form of the molecule changed. And after that, there's a whole complicated series of metabolic reactions on vitamin D. The big question has always been, what happens after the pre-vitamin D is synthesized? And why doesn't it show up in the bloodstream? And the answer is it does. This is another new paper. This is coming out of Poland, and it's on cryotherapy. So not cold plunge, but
Right.
whole
Right.
body cryo chamber. They took women, about 24 women in each group, and they compared those without multiple sclerosis to those with multiple sclerosis. The women without multiple sclerosis participated in the cryo chamber, no increase in blood serum level vitamin D. But the women with multiple sclerosis did get an increase in blood serum level vitamin D.
That is so awesome, Thomas. I mean, like,
Astounding.
I'm so happy to
We
hear you
now
say
have
that
this.
because again, for me it had been like a crude speculation or even that one with you and Max, and now to know that that's confirmed. And I wonder what the difference between the phenotypes would be between the non, MS patient versus the MS patient, why they would have a boost where they would not. Now, kind of along those same lines, you were talking about being in upper state New York and autoimmune diseases being more rampant there. You know, I know you know this, but I did a whole video on this where I looked at pretty much every disease process, and there's definitely like a U shape where like the lower the latitude, the lower the disease incidence of those other, and that could be cancer, that could be hypertension, that could be cardiovascular disease, Alzheimer's, et cetera, including autoimmune, rheumatoid arthritis, et cetera. So that makes sense. But so maybe those people had a sufficient vitamin D, and it just, I don't know. That's interesting to feel like there would be two different responses, I
No,
guess.
Casey, every single autoimmune disorder, every single one is associated with disruptions in vitamin D metabolism. This isn't just my son's story. This isn't just that longitudinal study in Finland. When you think about the formation of the immune system, because it's happening in that first year of life, and because vitamin D is essential to its development, even if you restore the vitamin D status of the individual later in life, the trajectory of their immune system is already cast by
Miss.
that first year. So my son wasn't necessarily vitamin D deficient at the time of the onset of his type 1 diabetes, he was deficient during the development of
Right.
his immune system. Parkinson's, rheumatoid arthritis, multiple sclerosis, even allergies, all of these other autoimmune disorders are associated with disruptions in vitamin D metabolism, but they're very difficult to detect because you might have an MS patient or a Parkinson's patient and evaluate their vitamin D and say, oh no, they're normal. You have to go back decades, like the Finns did with type 1 diabetes, to find the origin of the autoimmune disorder that only shows up later. Now, vitamin D status is still important to people with autoimmune conditions like multiple sclerosis. There's nothing that is going to bring back my son's islet cell function in his pancreas. Those cells have been destroyed. And even if you transplant from a twin new islet cells that make insulin into my son's pancreas, his immune system will attack those new islet cells and destroy them too. But not all autoimmune conditions are so irrevocable as type 1 diabetes. You can ameliorate some of the symptoms of Parkinson's or fibromyalgia or rheumatoid arthritis by repairing the vitamin D status, by sort of rescuing a vitamin D disorder. So, what might be happening in those multiple sclerosis patients in Poland that experienced an elevated blood serum level of vitamin D? We know that their bodies are not processing vitamin D the same way that people without the autoimmune condition are. It's quite possible that the vitamin D is made inside their brown fat and immediately released into their bloodstream because it is needed in their bodies in a way that is different than someone who isn't experiencing an autoimmune disorder. It's quite possible that they should do more cold. They should get more sunshine. They should go for further elevation because their body is so in need of additional vitamin D to try and correct the autoimmune condition that they're suffering from. There are some evidence that multiple sclerosis can even be reversed, which of course your profession would profoundly deny. There are
Profession,
something like
yes. Me,
seven.
no. I believe it. I think that the difference between a lot of the people that you have had contact with, I've seen again many podcasts with you, Nasha Winters, et cetera, is that the people who are not conventionally trained generally believe that the body has the ability to heal itself. Whereas
Correct.
the
You know
conventional
what
people
it
believe
needs.
that it's broken and we have to fix it for you, which is not true, but that's what they believe, right? So I do believe that. I think a couple things I wanted to touch on here. So kind of like when you're stimulated with infrared light and your body makes melatonin, do you believe that because you mentioned brown fat, but do you believe that it's anywhere with the mitochondria that's exposed to cold, or do you believe that the vitamin D is only being made in the brown fat portion?
I think anywhere. Let me qualify this. Your muscles are capable of non-shivering cold thermogenesis. Shivering is great. Shivering will clear glucose from the bloodstream. Shivering is a great way to make heat. But as you become acclimated to the cold, you don't shiver. You don't feel that urge to shiver quite so readily. Why? Because you have non-shivering cold thermogenesis mechanisms. Brown fat is one of them, but muscles will do it too. I talked to Denis Blondet at the University of Sherbrooke in Quebec, and he corrected my thinking on this. He said the quantity of brown fat in the baby is quite sufficient to keep the baby warm. Their muscles are not mature enough to shiver. And we lose brown fat, especially relative to total body weight as we get older. The quantity in an adult is insufficient to really explain non shivering thermogenesis. He says a lot of that can happen in your muscle tissue, and that sounds fine to me. However, wherever you activate the mitochondria, they will produce light. But where are you going to find the cholesterol? Where are you going to find the fat that the pre-vitamin D and the vitamin D will be stored in? Now, I'm guessing because I work mostly outside the body, not inside, but right in that fat cell, that's where you'll find the cholesterol. That's where the vitamin D metabolites will be stored. If it's in the muscle, well, I don't know what the concentration of cholesterol is. I don't know whether it's going to stay in I'm not as familiar with the chemistry of the muscle cell, but the biophotons will certainly be produced even by exercise. Here, I'm talking about cold thermogenesis, which in particular activates the brown fat. And it was a cryo chamber, after all, in the Polish study. I wonder if we were to, oh, I don't know, exercise women with multiple sclerosis, could we observe ultra weak biophotons, which they never tried to measure in the Polish study? Would that have a vitamin D effect? And I'm guessing, this is speculation, that it'd be less effective than activating their brown fat because of what's downstream of the biophotons.
Do you believe that the different frequencies of biophotons depends on the environment or activity that person is doing? Is that what you're trying to say? So
No,
exercise may not release UV biophotons and maybe more in the infrared, range or something like that, particularly?
no, I don't think so. Now, I would be amazed if there were not differences between the biophoton spectra in production of ATP and production of heat, because those are two different chains. But here we're talking about basic chemistry. When the electron moves from one valence state or one shell to another, that movement, as you know, it's not continuous. It's quantum. That is, it must jump from one state to another state. And as part of that jump, it will release biophotons. It will make light. We have no choice. It's the same thing. I don't know if you were in high school chemistry lab and maybe your high school chemistry instructor didn't do this experiment for you, but mine was a chemical engineer and he loved these sort of exhibitions. He would burn different metals to show us what the colors of light were. And so, sodium is orange. That's boring, but magnesium is white. And I forget which one was red and cobalt was blue. Now I can't enjoy a fireworks show without seeing the metals that are, being burnt in the fireworks. Copper is green. Each electron jump is associated with a different wavelength of light. And because these metals have their unique electron shells, when they're oxidized, of course, the electrons are jumping between different shells and different wavelengths of light are created. The electron that moves must emit light. It has no choice. And so the frequencies that are released as a result of mitochondrial activity are not sensitive to whatever stimulated that activity. They're sensitive to which electron shell is being, or sorry, from which cell is the electron being transferred and to what other shell.
Interesting. That's very fascinating.
ATP and thermogenesis, they have different endpoints, and it would make sense that they have different wavelengths. I have not examined the data to see whether one excites UVB more than the other.
Interesting. Oh, thank you for that. I appreciate that. I think that what I'll say is that you know, it's fair to say that cold does a lot of amazing things for us. Since structure equals function in biology, mitochondrial shape changes, organization, mitochondrial cardiolipin synthesis, super complex formation, autophagy, mitophagy, mitochondrial biogenesis, all of these things, brown fat, AMPK, that is, although extremely fascinating for me, and I would love to talk about it more. What I'll tell you is that even though I consider myself a mitochondriac, because Dr. Cruz had coined that term, black swan. I don't think I'm a black swan, but I'm definitely a mitochondriac. This channel, however, has been mostly talking about cancer being a mitochondrial metabolic disease. And so, when terms of talking about cold, the way I kind of wanted to at least pick your brain about it, because it's frustrating. The literature is very vast, but it's also so limited, and it's so frustratingly limited because the questions you specifically want to ask are not there, but then there'll be 10 questions you don't care about that are talked about, right? So within ketogenic metabolic therapy, as described by Dr. Thomas Seager, one of the core foundation is to lower blood glucose through the use of a ketogenic diet. And although that is a great foundation, what I found by working with people is that a ketogenic diet, to the degree that it needs to be with the glucose ketone index, et cetera, is fairly difficult to achieve um, on for a lot of people. And I don't know if you can
Why?
hear. Can you hear my dogs in the background?
No.
Okay,
But
good.
why
Because
is a ketogenic diet fairly
well, I
difficult to
will tell
achieve?
you because many people who have cancer, um, they are not metabolically well, to start with.
Correct.
If they were
Yeah,
metabolically well, they would likely not develop cancer. So
correct.
there's like there's the starting state of where they're at, right? And then number two, I'm not sure how close you follow Dr. Seyfried's work, but the GKI that they talk about of being at least less than 2, but ideally 1, is very difficult to achieve. That means your millimolar ketones are basically the same as your millimolar glucose. And
That's
that,
not a reason why, Casey. Why is
well,
that difficult to achieve?
well, I would say that it probably depends on the person. Everybody's their own equal n equals 1, and I've put together like, giant, PowerPoint resources trying to help people with this because I want to figure out any possible way that I can get there. It could be hormones, it could be vitamins, it could be, stress, it could be light environment. Many things can inhibit you from getting there. But what I just have to tell you that after working with a lot of clients who are trying to implement this foundational principle into their life, it just ends up being a difficult task for them, and they get demoralized because Seafried's like, well, look, you got to do it, you got to do it, and so
He
what I
probably sees it as a compliance issue. Seafood probably understands the dietary constraints that a person must conform to in order to ensure that they get into ketosis. But now you're talking about the glucose to ketone ratio. So you say GKI, I think
yes
you
sir,
call it.
yes
You
sir,
take your glucose reading, you divide it by your ketones. It's not just ketone, but it's also limiting glucose. And you are correct. The people who are metabolically dysregulated are going to have to go through a difficult period of transition. That is, they're typically too much insulin, typically hyperglycemic. Even if they get their ketones up, they have to also get their glucose down. Diet is only one way to do this,
agreed.
but when you're talking about GKI, it can take weeks. People will get discouraged, and then that Oreo cookie starts looking good. Like they get a little short-term comfort. So one way to think about this is they're failing to comply with the doctor's instructions, and that can be super frustrating for a clinician. But it's not the only way to think about it. One of the reasons that they fail to comply is even if they have a doctor who understands ketosis, there is so much information available on social media or chat GPT or confusion. For example, people think carnivore is keto. No, it's not. You can't eat all that meat and expect that you're going to get your glucose down because the amino acids in the protein become the substrate for gluconeogenesis, or maybe it's neoglucogenesis, for the
You're right the first
formation
time.
of glucose from essentially these amino acid substrates. People misunderstand this because their nutritionists and their dietitians lie to them about it. Now, maybe lie is an exaggeration, but when I met with my son's endocrinologist and I had the journals and I had the blood sugar data, this was before continuous glucose monitoring. And I said, look, we've got him on 0 carbs. He had a cheeseburger with a slice of American cheese. He doesn't even eat the ketchup. And look at what happens 3 hours later. I've got to give him more MPH. I've got to give him a little bit of H because he's getting up into the 200s on 0 carb. They're like, well, that can't be. They'll tell me, oh, that's not happening. Protein cannot be metabolized into glucose. Now I have to get a new endocrinologist because the data is sitting right in front of me.
Right.
When the clinician is so quick to blame the patient for noncompliance, they shut down avenues of insight into that patient because that endocrinologist thought I was lying to him. And it's not the first experience that I've had with a doctor that says, oh, this Professor Seager, he must be lying to me about what he's telling me. I had to discover that gluconeogenesis was a thing, that your body, even with 0 carbs, will maintain sufficient glucose to keep the synthesis of GABA in the brain going, to keep those processes where glucose is used as a material substrate, if not just for energy. Your body knows how to do this. And if you overload it with protein, it will create more glucose. Carnivore and keto, not the same thing. And so a patient might think that they're complying because, they read a well-meaning, I don't know, Sean Baker is great. He's a medical doctor. And he's saying, look, carnivore is helping me, but carnivore is not ketogenic.
Right.
And Baker knows this and he doesn't confuse people. Other sources of information get people up in their mind about what they're trying to do.
I think that's a great
I'll
point.
let you get a word in edgewise.
No, I think that's a great point because it goes back to it's not all about diet. Dr. Cruz talks about this. Blue light exposure right now, I'm being exposed to probably 400 nanometer light, 450, just so it looks decent on camera. That is going to raise my cortisol, my catecholamines. That will make gluconeogenesis happen in my liver. And that stress, is a real thing, and that stress can come from a lot of different
Correct.
sources, right? So I guess that's what I'm trying to get at is that yes, I want to make sure their macronutrient ratios are like that makes sense, right? But I want to give them as many tools as I can to get their glucose ketone index down without getting frustrated. And that's where I wanted to talk to you about cold. And this is where I was trying to say that there are becoming more studies. They're publishing more studies on cold because it's becoming a more popular thing. But there's still frustratingly less a lack of studies for the questions that I particularly want. And I just want to know if you could help me shed some insight on that.
We don't need more studies. We have all the information that we need to act. Now, I'm not arguing against them, like studies are interesting, but we don't need more research to say, oh, let's get the cancer patient into the cold. Cold stimulates production of ketones immediately. You get in there and you begin to clear glucose from your bloodstream as soon as you've activated the sympathetic, nervous system. There's some glycogen release from your liver, especially if your, glycogen stores are high. And that's great because that's part of the sympathetic activation. And then cold thermogenesis will begin clearing that glucose almost immediately. Denis Blondet tells me that most of it's in the muscles, some of it is in the brown fat, and it's fine. We have continuous glucose monitoring data that will show a healthy person, no diabetes, will spike up to about 125 or 140 as soon as they get in. 30 seconds later, they're back down to 90. You might say, oh, that sounds stressful. Of course it's stressful. There's a cortisol release, especially if they're not acclimated to the cold. A lot of things are happening hormonally, and this is a hormetic stress. Your white fat cells will begin to release fats into your bloodstream because that is the principal fuel of non-shivering thermogenesis in brown fat. And so anybody who's been cold training for a couple of weeks is recruiting new brown fat into their body, improving their insulin sensitivity, improving their metabolic flexibility, and generating ketones almost the moment that they get into the cold. So you can use your cold and your light environment in addition to your macronutrient makeup to keep yourself in a state of ketosis and improve your GKI. That is, keep glucose down, which also pushes insulin down, and keep your ketone levels up. For those people trying to build the metabolic flexibility that they've never had, it does seem like it's a lot. Geez, you mean I have to fix my light environment? Yes, you do. Gee, do you mean I have to fix my thermal environment? Yes, you do. Do you mean I have to sleep? Do you mean I have to minimize the seed oils in my diet? Yes, you have to do all of those things. And you have to do it because you're trying to save your life. What Seafried misses is, although he gets right that cancer originates in disorders of mitochondria, what he misses are the mitochondrial therapies. There are multiple routes to mitochondrial injury. Excess carbohydrates, it's just one of them. There are when you starve the tumor cells the way that Seafried is suggesting, you will get better cancer treatment outcomes. But there are multiple mitochondrial therapies that anyone who is working on a mitochondrial disorder should be incorporating into their regimen. So let's talk about the routes of mitochondrial injury. Light environment is the first one. When your light isn't right, then your mitochondria will be exposed to reactive oxygen species that they don't have the melatonin to donate enough electrons, to absorb. Now, a little bit of ROS is fine. It's a bright day. I've got all kinds of blue light hitting me right now. It's no big deal. It's during the day. You need bright days and dark nights so that your mitochondria can produce the melatonin they need to protect themselves against excess reactive oxygen species. Second route, seed oils. People really underestimate how damaging these seed oils can be. Danny Jones just had Nick Norwitz and Paul
I
Saladino
saw
on
that.
a
It was a
podcast.
fantastic podcast.
Wasn't that fun?
I could not get enough of
Nick's
that podcast.
there's like 3 hours of it, you
It
know,
is.
and for your audience, I'm going to summarize it somewhat unfairly, but Norwitz is like, seed oils, they don't seem that bad, and Saladino's like, no, 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 they're very bad. Neither one of them really understands the mechanisms by which seed oils will damage the mitochondria. They're thinking about metabolism, and it's true that if your body is going to oxidize, I shouldn't even say oxidize, if your body is using the seed oils for respiration, it'll break all the double bonds. It's not a big deal. You can metabolize these things, it's fine. And so that sort of sounds like I'm agreeing with Norwitz, but I'm disagreeing entirely. Saladino was correct to point out there is no such thing as a linoleic acid deficiency in anyone living in an industrialized country. Yes, omega-6 are essential. Yes, linoleic has an essential role to play. And we are so overwhelmed with linoleic acids that we'll get plenty, even if we minimize seed oil consumption to zero. The real danger in the seed oils is not in their energetic implications, but in their material. They are
Correct.
incorporated into our cell membranes as phospholipids. And they do not, because the location of the double bond is different, they do not perform the transport functions in the same way as the omega-3 seed oils do. They become resistant to the action of insulin. They do not make the same kind of effective semi-permeable membrane when the phospholipids are made from omega-6 rather than omega-3 fatty acids. You get these proportions out of whack. And the brain is the first place to suffer because the brain is made up of phospholipids and fats, and in particular, DHA, which is an omega-3 fatty acid. I'm getting this from Stephen Cunnane, also at the University of Sherbrooke, who wrote a book called Survival of the Fattest. I never would have found Cunnane if it weren't for Jack Kruse saying, hey, you should look at these books Cunnane is, writing about the development and physiology of the human brain. The human brain requires DHA. And it's most important in utero and in the first sort of 3 years of life where the brain is developing. If it does not get the omega-3 fatty acids, in particular DHA, that it needs, and we know fish, shellfish, aquatic sources are the best for this. They also come with the iodine that the thyroid needs. If it doesn't get it, it's no wonder that your brain and your entire neurological system will develop along some aberrant or atypical trajectory. And now I'm going to make a leap for you, Casey, because it is a big issue. What causes autism? And Kennedy comes out and says, oh, it's the Tylenol. And by the way, you can use folate therapy to help treat autism. There is a strong association between Tylenol and autism. There is a strong association between every pain reliever, fever reducer, and autism. You know why? Because it's not the Tylenol. Tylenol's toxic. Don't give it to pregnant women and don't give it to infants. But it is the fever. It is the immunological event that the parent is trying to treat with the Tylenol that explains the association. It is mostly a confounding factor. How did my son get his diabetes? It
That's
started with a
fascinating.
fever, Casey. It was his immunological development was abnormal in his first year, but he was 6 when the diabetes first showed up. And that onset was initiated by a fever. Now we're going to go by analogy. So this is speculation. An autistic child, we know, has an atypical neurological system. The brain isn't working the same way. The whole nervous system is off. They have sensory processing issues. We understand all of this. 80% of the autistic children between the ages of 2 and 5 in this California study showed markers of mitochondrial dysfunction. The greatest risk factor contributing to a diagnosis of autism in a child before their 10th birthday is the metabolic state of the mother during pregnancy.
Fascinating.
It's not the Tylenol per se. It's not the microplastics. It's not even the vaccines. It's not all of these things. I mean, it is.
It's everything.
Autism is a multifactorial disorder,
Exactly.
but they all act through the same pathway, which is the mitochondria. But it is not until an immunological event like a fever or a vaccine tips the scales beyond the brain's capacity to compensate for this mitochondrial deficiency. When they are under that additional stress, for example, encephalitis, the swelling of the brain associated with the fever or the vaccine, that is when we see neurological regression. Now, if this is the case, and it is, then would you expect therapies that target the mitochondria to improve the neurological capacity to recover some of these neurological functions and treat autism? Of course you would. This is the proof of the hypothesis. Folate therapy is a mitochondrial therapy. The question is, why are these mitochondria so dysfunctional in the first place? We talked about light. We talked about seed oils. We can talk about excess carbohydrates. If there's excess carbohydrates with no break, no ketosis, no fasting, then the mitochondria will never recover from the reactive oxygen species required to process all of these carbohydrates. And then the fourth one, the route to mitochondrial injury, is environmental toxins. I mean, Saladino's right to say you don't need the antimony that will leach out of the plastic bottles into your seed oils. You don't need lead. You don't need mercury. There are some things that perform no function in the human body. One of them is ionic aluminum. It should stay the hell out of your diet. It's okay that aluminum is in the dirt and in the silt when it's bound up with the silicates and it's never really let your kids eat dirt. It's a lot of aluminum, but none of it is released into the body. It is when the aluminum is added at the water treatment plant in the form of aluminum sulfate, and that's an ionic form. It's more mobile and it becomes absorbed into the body. That's when it's neurotoxic. And the addition of fluoride only makes it more mobile and available. So those are the four routes. It's the light environment, non-native EMFs in particular, the seed oils, the carbohydrates without end, with no break, and toxins. So where does the cold come in? Cold helps repair all of those. Cold is another hormetic stressor, but it stimulates mitobiogenesis. It is a mitochondrial therapy. You will recruit new mitochondria into, for example, your brown fat cells as a result of cold exposure. Here's the next mind-blowing fact, and this is, again, very recent. I got this from Martin Picard at Columbia University.
I watched your podcast with him.
When you recruit, he'd be a great guest for you. When you recruit new mitochondria to your brown fat will release mitochondria into your bloodstream.
That was fascinating.
Mitochondria move throughout the body. And there's a paper that Picard cited for me. I had to go look it up. In one case, fat cells released mitochondria into the bloodstream that were transported to damaged muscle cells at the heart and repaired the mitochondrial function there. So I know I'm on a long rant. I'm a professor and I'm lecturing Casey. I'm going to wrap this up here. Seyfried is correct to suggest ketosis. He's correct to say, look, these cancer cells, we're going to get them where they're weakest. We're going to get them at their metabolism. We're going to starve them of the substrates to fermentation, glucose and glutamine that they need for energy. But what he misses is, how do we fix the mitochondria in the first place? If we can bring the mitochondria in that tumor cell back online, then it can signal apoptosis and destroy the tumor cell. Wouldn't that be wonderful? Cold produces new mitochondria, and this is all speculation now. It is possible that some of those mitochondria will travel to the tumor site, and at least in the edges of the tumor, the cells that are most exposed, replace the defective mitochondria, restore mitochondrial function, change the redox environment around the tumor, signal apoptosis, and cause the tumor to shrink. If that is the case, then we would expect to see people who incorporate cold plunge therapy into their cancer therapies. We would expect to see their tumors shrink. And we do. Writers are sending me imaging data. They're sending me blood markers, and they're saying, I started ice bath therapy. and now my tumors are shrinking. This is the best evidence yet, I think. I mean, it corroborates the Secchi study on the
Hmm.
mice that show
Correct.
ketosis and cold will inhibit the growth of tumors, but it's the best evidence yet that there's something going on in the mitochondria of the cancer cell that can be repaired by these mitochondrial therapies. That's a good place for me to stop and
Well,
allow
I'll
you
tell you
to.
that that has really stimulated a lot of thoughts and questions. And that's probably one of the things that I most admire about you is that you are so freaking curious and you have such great ideas. And, I've heard you say this on multiple podcasts, but it's the ideas, it's the hypotheses that are the fun part about science, number one.
That's right.
Number two, it's just such a stark contrast from my profession, which is so uncurious, so burnt out, so this is the way it is. We feel like we know everything. The science is settled, and there's no such thing. And it's just frustrating, to be part of that sometimes. But what I'll tell you is that I had a great mentor when I was right out of residency. I did a functional medicine fellowship, and the guy was a chiropractor, but functional medicine doctor. He worked with the IFM, and he distilled basically disease down to uh, four main mechanisms. And what he said, he called them the four horsemen of the apocalypse. I'll never forget this. And it was insulin resistance, catabolic physiology, which is basically a disordered, cortisol axis, um, oxidative stress, and chronic low-level inflammation. And I will tell you that when you
Those are
study,
all like the same thing.
right, but what I'm saying is when you study how toxins or how hyperglycemia or how stress functions through the body, they all work through inflammation, through oxidative stress, through disordered, hormone physiology. But the way that those, and we can detect those very easily in the bloodstream. I can do a 4-point cortisol, whether it be salivary, urine, blood, you name it. I can check all your testosterones. I can check HSCRPs and oxidative stress markers. That's fairly simple for the clinician who's curious and looks for those things. But the real question is, where did it come from, right? And that's where you're looking at all the different factors, and that can be different for every person. The second thing that stimulated me was the fact that, yeah, Dr. Picard's podcast with you was so fascinating, talking about the mobile mitochondria, how there's mitochondria in the bloodstream. That was so cool. And I had seen papers of there were like neurological studies where they took like basically the neurons that were defective in like a Parkinson's or Alzheimer's disease model had like a bridge, almost like an axon, like a nerve that would like bring over the mitochondria. But I wasn't aware of this free floating mitochondria. Number one. Number
No.
two, I mean, um, a couple of things. Mitochondria, from my understanding, one of the things that from if you look at Seafried, the way he talks about killing off cancer cells, it's leveraging the fuels, glucose and glutamine, but then hitting them with oxidative stressors. But there's a lot of other metabolic therapies that make a lot of sense. One of them, which is like dichloroacetate, DCA. And what it does is it basically funnels electrons or energy flux back through the mitochondria that's broken, and then the ROS signal then will trigger apoptosis. So what you're saying makes sense too. Maybe there could be a portion of those mitochondria that could be shuttled in, potentially, and then retake over metabolism and the kind of the protective mechanisms, and if it's too far gone, then reinitiate apoptosis. I guess I always thought about cold as just being a shunt of glucose. I was reading that paper that from the applied physiology paper that you have on your website, but I'd read it before, where it was like 80% of the energy used by cold is glucose, right? So I'm like, well, it just makes sense that it could just be glucose, but you're giving me another potential mechanism that could be going on too, which I think is very fascinating. Um,